Hello, I'm Dr. Ray, and today I'm going to talk to you about neurulation. Now, neurulation was introduced to you in the week 3 through 7 video that I have, and so I'm kind of just going to keep going where I left off, where I introduced it to you. What is neurulation? Neurulation is a process where you have first a neural plate form, and then the plate is going to actually create a tube, and then we're going to call this tube the neural tube, which is going to develop into the brain and the spinal cord. And this process is going to occur throughout the third week, or it's going to start in the third week, and it's going to finish at the end of the fourth week when the ends of the tube are going to close off. So this is a dorsal view of an embryo. You can see the somites here on the dorsal side, and it kind of looks like a long cylinder from this view, and you can see that part of it seems to be closed, or the ends are coming into close approximation with each other. So that's the neural tube there. So this process of forming this tube is called neuralation. And the main player in this process is called the notochord, or the main player that is going to induce this process, rather. So here you can see the notochord. I talked about the development of the notochord in my video on weeks 3 through 7. So the notochord is in the midline of the embryo. This is a transverse view through the embryo. You can see the surface ectoderm and the endoderm here, and the notochord that's developing in the midline. The notochord is developing from mesenchyme that came through the primitive streak. Here you can see embryonic, intraembryonic mesoderm depicted here. And so the notochord is going to actually induce thickening of the ectoderm that is overlying the notochord. Okay, and so here we're going to call that part, you can see it getting taller here in this little animation, we're going to call that thickened area the neural plate. And so the notochord is going to induce the thickening of the ectoderm to form this neural plate. And this special ectoderm that is now thickened, that make up the neural plate, is referred to as neuroectoderm at this point. And so that's essentially just what we refer to that region. And that neuroectoderm is going to develop into all of the central nervous system and other structures, like, for example, the retina. So when you do development of the eye, you'll understand how this isn't only going to become the spinal cord, but there are some other structures in the body that it's going to form as well. Now, at first, the neural tube and the neural plate is going to be as long as the notochord. So in this picture here, you can see depicted in red is the notochord. So this is the sagittal section through the embryonic plate. And it's trilayer. Here's the endoderm. And if you remember, during development of the notochord, the floor is going to degenerate for a period of time. And so here you have the notochord developing and the thickened neural ectoderm overlying that. And so they're going to be the same length at first. But the neural plate is going to overgrow the length of the notochord and is going to extend beyond it. And we talked about that in the weeks 3 through 7 when I talked to you about the head fold and the tail fold that occurs during folding of the embryo. So the neural tube is going to grow rapidly. And this is also a sagittal section of the embryo, but kind of a big picture of the whole embryo that's showing you even the umbilical vesicle. And the reason why the neural tube looks the way that it doesn't look like an actual continuous structure, is because the neural tube that's depicted here is hollow. It has a hole in the middle, so it's almost like cutting a straw in a sagittal section, and that's what you're looking at there. The dark pink line is representative of the notochord. All right, so you have this neural plate, the thickened ectoderm, and the folds are going to start to grow taller, and the plate appears to invaginate along the midline. And we call this groove that shows up the neural groove, okay? And so this neural groove becomes very prominent here at the cranial end of the developing embryo. And this is going to actually be the first sign of brain development. So this is the region of where the brain is going to develop, and we'll talk about that at another time. So towards the end of the third week, these folds that are growing upward towards each other come in close approximation with each other, and it's going to actually change that neural plate into a neural tube. And then the tube is going to kind of seem to sink down into the embryo. It's more like the embryo is growing. And the ectoderm is going to come together, and it's going to fuse with the other side, and it's going to make a continuous surface ectodermal layer, just like we had before. Now, the neuroectoderm cells that are in the neural tube are going to change from epithelium. So they were an epithelial cell, which is a type of cell that have close junctions. They're closely adhered to each other, and so they don't migrate or move around very much. As you can see those depicted in this cartoon as being cells. I kind of drew these into your textbook schematic that they had, and that's going to undergo a transition into mesenchyme. And I talked to you about the characteristics of mesenchyme in weeks 3 through 7, but here I'm going to kind of give you a cute little animation where here we go, we're going to transition. And so instead of being closely packed cells with tight junctions, they're going to be more loosely arranged, pluripotent, and mobile, so they can migrate, okay? And they can change into a lot of different types of cells. All right. As this neural tube was separating from the overlying ectoderm, some of the neuroectoderm cells lose their epithelial affinity. So they do change from epithelium, but they don't actually become part of the neural tube. And so they kind of become positioned between the surface ectoderm and the neural tube. And we're going to call these cells, they're going to be destined to become neural crest cells. And so they're not going to be epithelium anymore, but they're also not going to contribute to the neural tube. And so you can see them kind of depicted here in this light blue color without a nucleus. And then they're going to move kind of into two different condensations on the dorsal aspect of the neural tube and kind of move more laterally. So there's going to be one on each side. And so it kind of seems like this is what's happening here, but in reality, it's a differential growth. So the embryo is actually growing bigger, and that's why the neural crest cell condensations seem to change their location. Okay. And so these neural crest cells are not just going to sit there. They're going to migrate and move into the developing somites that are part of the paraaxial mesoderm, and also the areas that are more superficial to the somites. So the neural crest cells are going to distribute widely in this region, but they are going to really distribute all over in the area of the embryo that's going to develop into the head and neck. And we'll talk about that in a minute. We're going to talk about specifically where neural crest cells go or what they turn into. Okay, so where do they go? Or what do they become? The first thing that the neural crest cells become, and when you think neural crest, I hope that the first thing that comes to mind is that they become a ganglion. So in this depiction, I have drawn a spinal cord, and I drew little pretend cell bodies here that have processes sticking out, and I'm trying to represent this dorsal root ganglion, which are also our spinal ganglia, so they're called the same thing. Neural crest cells are going to become these ganglion, okay? And so these cells that are derived from neural crest cells are responsible for directing afferent signals, so sensory signals, towards the central nervous system from the periphery. So the signal starts out here in the periphery, and then the signal is going to come in, and the signal is going to be handed off to the next neuron, that is a cell body that is going to be in the spinal cord in the central nervous system. We'll talk about that again. Where the peripheral processes grow out to, they grow out to lots of places, anywhere that's going to receive sensation. I guess the easiest thing to kind of hold on to is somewhere like the skin. So the skin is going to have these nerve processes that have different types of receptors at the end of them that are going to receive different types of signals or different types of be stimulated in different types of ways that allow these neurons to hand off the signal that will go up to the brain and will tell your brain that something is occurring in the periphery of the body. They also are headed to walls of organs and other places, but the skin is just one of those things that's easy to think about conceptually when you're just starting off. Where are the central processes growing towards? The central processes are growing towards the spinal cord. These central processes are going to become the dorsal roots of the spinal cord, and they're going to synapse on cell bodies, other neurons that are sitting here in the dorsal horn, and some of them actually come over and they go all the way up to the brain stem. Okay. Neural crest cells can also become other ganglion. We do have some sensory ganglion that are in the cranium that are associated with cranial nerves 5, 7, 9, and 10. And so those cranial nerves are thought to be partially derived from neural crest cells because the sensory ganglion that are associated with these cranial nerves come from neural crest cells. So they are synonymous with the spinal ganglion in function. There aren't synapses occurring in these ganglion. They are just where the cell bodies are for these neurons that are bringing sensory information towards the central nervous system. All right. Neural crest cells also become other ganglion, like autonomic ganglion. Autonomic ganglia is a ganglia where you have cell bodies that belong to neurons, but there is a synapse actually occurring in this one. So autonomic ganglion are different than spinal ganglia and cranial ganglia. So here you can see I drew in these little pink blobs. Those are supposed to be the cell bodies of neurons. And then you can see axons coming in, and these axons belong to other neurons that are within the central nervous system. And those neurons, axons, are coming all the way out, and they're handing off a signal, they're synapsing with the cell bodies of the neurons that are in these autonomic ganglion. So the neuron that have these axons before, we call those presynaptic fibers that are coming through, presynaptic axons. And they're coming in and they're going to synapse on these neurons, and we call these postsynaptic fibers here. And so they are going to places, and you'll learn about where they're coming from specifically and where they're going to later on in this course. But these ganglion themselves, these cell bodies and these axons that are growing out from them came from neural crest cells. But the pre ones didn't, because they're growing out from the central nervous system. They're not actually growing from the ganglia here. Neural crest cells also contribute to or make up the neural lemma sheaths that are in all of the peripheral nerves in your entire peripheral nervous system. So all of the Schwann cells came from neural crest cells. So if you think about any nerve that's in the peripheral nervous system in your body, they are wrapped by cells that came from neural crest. So now that kind of helps you see how widely distributed a neural crest cell is. And then they also make up the leptomeninges, which is a name that means pia mater plus arachnoid mater. So, but the pia and arachnoid are very closely approximated to each other, and they actually have trabecula that span between the two. And so it's not hard to realize that they came from the same tissue. So they actually came from neural crest cells. The dura mater that's on the outside did not come from neural crest cells. That comes from mesoderm. And so that one is not a part of the neural crest cell derivative. So that's why we call these two the leptomeninges. And then neural crest cells also become all of the pigment cells that are in the entire epidermis. We call these cells melanocytes. They make melanin, and you'll understand when we get to the skin in histology that the development and production of melanin is very similar to the production of a neurotransmitter, and so it'll help you remember that these cells all came from neural crest. And then also, this is my cartoon drawing of a kidney. And here you can see the suprarenal or adrenal gland situated on the superior pole of the kidney. The adrenal gland has a cortex that has layers, but it also has a medulla in the middle. And the special cells in the medulla are called chromaffin cells, and those cells came from neural crest origin, and you'll understand they make neurotransmitters, how that's all connected later on in your training. Now, in the head and neck, neural crest cells can essentially become almost anything, including connective tissue. So I'm going to kind of break this up with sort of a silly little animation, because I just said that in the head and neck, neural crest cells can become anything, even connective tissue. So usually connective tissue comes from mesenchyme. Oftentimes it's mesodermally derived mesenchyme, but in the head and neck, the mesenchyme can be derived from neural crest cells. And so that makes it different. That's different than the whole rest of your body. So in this case, in the head and neck, neural crests can become things like bone, things like muscles, things like other types of connective tissue that you'll learn about what that is later on. But okay, so I just want you to hold on to that pattern here. All right, so the summary of neurulation. The neural tube is going to come from and be stimulated by the notochord. I said that kind of not the way I wanted to. The neural tube is going to be induced by the notochord, so it doesn't actually come from it. I shouldn't have said that. It actually comes from neuroectoderm. But this neuroectoderm is going to differentiate into all of the central nervous system. The neural crest cells that also come from neuroectoderm are going to give rise to a lot of the cells of the PNS, because it's going to be all of the Schwann cells. And it's also going to give rise to ganglia, especially the autonomic nervous system ganglia that you'll learn so much about throughout your training. But it also gives rise to all of the dorsal root ganglia and the cranial ganglia. The cranial 2/3 of this tube, this developing tube, is going to develop into the brain. And so if you watch the movie on brain development, then you'll get to carry on with that. But then the caudal 1/3 of this neural tube is going to develop into the spinal cord. And I will have a separate movie on the spinal cord.